Welcome to the SwineTime podcast here at Pipestone. I'm your host, Spencer Wayne. I'm one of the staff veterinarians here on staff, working with farmers. I'm also one of the owners in Pipestone Holdings. And besides going out in the field and working with producers, the one cool, fun thing they let me do every so often is to host this podcast. And I get to interview different people. A lot of them are folks that work within our company that are doing interesting things, but on occasion, and as often as we can, we get folks from outside of our organization. And today I have just such a person. Dr. Kim VanderWaal from the University of Minnesota is going to join us. And Kim, I'll refer to as Kim. I already asked if she was fine with that, and I wouldn't offend her by not calling her Dr. VanderWaal, but she's going to talk to us about PRRS classification and how we describe the viruses that we work with today. So Kim, I'll turn it over to you. And why don't you just take us a little bit through your own life and your background and how you came to be where you are now at the U of M, and then what you do there.
Sure. And first, thanks so much for inviting me to be part of this today. It's always great to be able to have an opportunity to reach our stakeholders in Minnesota and beyond. So as you said, my name is Kim VanderWaal. I'm an associate professor at the Department of Veterinary Population Medicine at the University of Minnesota. I'm from Minnesota. I grew up in Bloomington, and I did my undergraduate here at the U. After that, I did my graduate education at the University of California at Davis. And then was lucky enough to be able to come back here and eventually start my current role as on the faculty here at the College of Veterinary Medicine.
Okay. Hey, you just bounced through a few things. Did your graduate education at Davis? Did you go to vet school? I assumed you were a veterinarian.
That is a common assumption because I do work for the veterinary college, but I actually have not trained as a veterinarian and have a PhD. So I come to this field from the perspective of disease ecology and epidemiology. All
right, I have a newfound respect for you, different than I had before. Now that I know you didn't have to go through all the mucky education that I did on this, so you're a smarter person than I thought.
Well, I have respect for people who did that veterinary degree. I guess you're a really broad background, a broad basis to understand animal diseases.
Okay, good. All right, so you're at the U of M now, and what's your position, and then what do you work on?
So I am an associate professor. I have been on the faculty since 2017, and I've been working on viruses of swine since about probably 2015 at this point, so 10 years. And the main area of research that we are interested in studying is how viruses spread in the population, in animal populations. And here, of course, we're talking about pigs. And so a lot of the work we've done is on PRRS. We do study other viruses as well, like foot and mouth disease and porcine epidemic diarrhea virus. But we probably about 70% of our research focuses on PRRS. And the reason for this is PRRS is obviously a huge issue for the swine industry in the U.S. It's also a really interesting disease to study because we have so much data. It's one of the most sequenced viruses in the world. And so we have the level of granularity we have on which strains of the virus are occurring where across the landscape and across time is almost unparalleled to any other virus that you can think of, including ones that affect humans, right? So it's a really interesting virus to study, not only because there's real problems that we're trying to address, but also because I think that we can learn something about viruses and their epidemiology by studying these viruses in pig populations.
Yeah. So I know you, in my mind, you are a big data person. You were doing big data type studies. When I say big data, I don't mean like just a generic, like it's big data. I mean, it's the ability to take large swaths of data and try to use sophisticated tools to pull truth from that or interesting facts from that you wouldn't normally get if you just did a cursory analysis of it. Is that pretty accurate of what you were doing in general terms, or how would you describe it?
Yeah, I mean, it's somewhat what we do. I would say that I am a data integrator. I like to pull data from different types of places and put it together in novel ways to get answer, both old questions and new questions, and try to do it in ways that really take advantage of the rich data landscape that we have in the pig industry. And PRRS is a great example of big data. We have probably 80,000 sequences that have been generated. So these are genetic sequences of the virus generated from the US, which is a huge number. And it does require a lot of big data type tools to be able to actually make any sense of what's going on when you have that amount of data through time.
Sure. Okay. So I always want to keep one thing in mind as we go through these things, especially if two data-oriented people start talking. A lot of our listeners are farmers, most actually are. And so just to kind of set the stage a little, we're talking about PRRS. Genetically, we're talking about looking at the genetics of PRRS. PRRS itself, if you think about base pairs, I had to look it up before we started. I've got about 15,000 base pairs total. So if you think about each base pair is a gene, and every 3 genes flip on a protein, or not a gene, I should say, it's a base pair. Every 3 base pairs codes for a piece of a protein. But anyway, the genetics would be 15,000 A, T, G, or C long. And is that right? Am I right on that?
That's correct. Yep.
And then there's about 600 of those that's in a part called ORF5. And out of the 15,600 are a little special set of genetics that we have traditionally used to classify our viruses. We'll look at that ORF5 sequence and say, oh, we would have called it things like a 144 or a 132, a 252, 111. There's all these different codes that we have given it, kind of like a little license plate. But we're changing that now. And so as we talk about the genetics, we got 15,000. We're looking at 600 of that 15,000. And we called it one thing in the past, and now you're proposing a different way of calling it. More than proposing, we've already switched to it. So can you describe how the old system worked and why this new system of classifying viruses is better and how it works?
Yeah, I'd be happy to. So I want to step one step back first for your listeners, and we're talking about the sequences that we generate from the virus from farms. And so if you're a farmer or a veterinarian and there's a new PRRS outbreak on one of the farms you manage, often what is done is a sample is collected and it is sent into one of the diagnostic labs. And there's basically 2 things you might want to know. There might be more than things, but 2 of the things that you might do is first say yes or no, do the pigs on this farm have PRRS? And second, well, which PRRS they have. It's a very diverse virus, and often the reason why we would want to generate a sequence is we want to know, well, is the current strain that we have the same or different than what we had on the same farm last year? Or maybe this is a strain that's already circulating in our system, or maybe our neighbor has it, or maybe it's the vaccine and we're just sequencing the vaccine virus. So those are the types of things that we might want to learn from generating a sequence. So we do have historically, and actually, all the way back to the 90s, we sequenced that OR5 bit of the virus and use that as kind of our license plate, right? And the naming scheme that's historically been used is the RFLP type. And this is a tool that was really based on technology of the 1990s, and you get to this sort of label. It's a 144, it's a 174, it's a 124, all of these sort of basically 3-digit names we have for the virus. But the issue is that RFLP types don't work very well. And what I mean is that you can have 2 viruses that are very different. They might be 10% different from each other genetically, but they will receive the same RFLP label. And that's not useful because now you're getting this information and it's telling you 2 viruses are the same and they're actually very, very different. So it's giving the wrong information.
Yeah.
You can also have...
Go ahead. The illustration of that is exactly what happens when you're working with a farmer or let's say there's sows involved, because that tends to be something we study a lot on the virus repeatability. And you'll have a 144 and it's a 144, and then you'll find a 124. And then typically the person in the barn will say, hey, it's not the same. You say, no, it is. It's 99% the same. It's the same. It flicked one little molecule off and now it's a 124. And that is a hard thing to get past, because if you kind of know how it works, you can understand it. But if you're just hearing somebody talk about 144, you think 124 must be something completely different. No, it's the same virus which is why that naming standard or that way of identifying viruses is not terribly accurate. You can get confusing pretty quick if you start using those only.
Exactly. You get both problems. You get different viruses being called the same thing and viruses that should be called the same thing receiving different names. And it's just a recipe for confusion and hasn't worked very well. And when I really started getting into looking at genetic data or into the PRRS virus, this was probably about 8 or 9 years ago. Well, we said, well, we have RFLP typing. We can't do any better. This is just what we have to deal with. And eventually, I sat around and I thought about this and watched it for a few years and I said, well, why can't we do better? This is not the 1990s anymore. We have better tools. We have better computers, and we have a lot more industry cooperation to have these sort of national level databases where we get a good picture of genetic sequences collected from PRRS all across the country. So we have much, much better tools and opportunities now, and that's really what started us down this road of developing a better classification system.
Did you invent this new system? Was that you? Or I have no idea where the new naming standards came from, but it was you.
It was a initiative. I'll say that I led the initiative. The adoption of it was spearheaded by a working group that was under the umbrella of the American Association of Swine Veterinarians PRRS Committee. And so we had input from veterinarians as well as all the major diagnostic labs, as well as other researchers have been very involved with PRRS monitoring in the country, both here at Minnesota, but also some initiatives from Iowa State as well. So it was definitely not solely me. The system is also an extension of the lineage-based classification that was proposed about 15 years ago. And I was still in grad school there then, and I wasn't studying pigs yet. So I was not involved with the initial proposal of lineages. And I'll step back here and maybe talk about what a lineage is, right?
Yeah.
For your audience.
Before we get into that, just to make sure, because I want to make sure everybody's kind of going along with us in the conversation. That little 600 base pair section we call OR5 used to be called things like 144. Wasn't terribly accurate. Yes. And then not just you just clarified, but you spearheading the effort. So there's a better way to call these viruses to avoid confusion. And that's this new system you're going to discuss with these variants. And I'll turn it back over to you now. And why don't you describe that a little bit?
Yeah, so there's two level of classifications now that we assign to these viruses, still based on OR5 gene. The first is we assign it to its lineage and sublineage. So that's kind of a larger ancestral group of viruses. And so these are, when you hear like lineage or sublineage 1A, 1C, those are all different sublineages of the virus. And so this sublineage system we adopted in, I think, starting in about 2019. Through some work led by who was a postdoc at the time, Igor Poplawski, was the one who started putting into place the current sublineage system we used, which has later been refined by Dr. Zong's group at ISU. And so those are allowed us to place these viruses into these large families. The issue for these is these family groups are really big, right? For example, right now, most of the wild type viruses that we see in the U.S. probably are belonging to just 3 or 4 of these big sublineages. So there was a need to further stratify those, because the things that practitioners want to know when they're sequencing viruses is, again, is this the same virus as we had last year, or is this different? Sublineage is not going to get you there, because that's a really big group and doesn't quite have enough, precision to get to that level. So the variant system then takes these sublineages and further splits them apart into groups of viruses, where at the sequence level on OR5, the viruses are around, on average, 2.5% from one another. So if you have 2 viruses that come back as the same variant, they are on average, 2.5% from one another. That's an average, so they might be more.
When you say variant, you said lineage, sublineage, and what is variant? Where does that come in to the? I'm thinking it's like group, smaller group, little lineage, sublineage. But what is variant? Where does that pop into in this?
So it's nested within a sublineage. So one sublineage is made up of numerous variants.
Okay.
And so, for example, in sublineage 1C, which is one of our big sublineages, we have variant 1C5, which is one of the most common ones that have really been an issue in the Midwest in the past, 4 to 5 years. But we also have other variants in that group, like 1C4. There's some 1C twos that are quite prevalent right now too. So these are all variants. They're all different subgroups within that broader sublineage.
Okay. So when you say, just for the producers who might be looking at these lab reports, 1C5 is lineage 1, sublineage C, variant 5. Is that the right way to say it? So
that is correct.
1C5 it's a lineage 1, sublineage C, 0.5, which is the variant. And you're saying right. If you say I'm a 1C.5, well, that will have a max of about 2% variation from one virus to the other. And essentially, it's the same, very, very similar virus or the same virus.
It's an average, right? It's an average of around 2.5%. So you will see a little bit higher than that, but on average, you'll see that those viruses are around 2.5% from one another. And they're usually about 5% different from the closest related variant.
Okay. All right. I don't have anything to add, so you keep going. What else do we know about this?
So the great thing about these variants is it's much more based on the biology, right, of the virus and the way that viruses evolve. So you're not going to have the sort of issue that we had with RFLP types where you have, say, a 1C5 break on a farm and you keep sequencing it over a few months and all of a sudden it changes its name. These variant IDs are much, much more stable than what we would see with an RFLP type. So if all of a sudden you see a different variant ID, then you can be much, much more confident that this is a new introduction of a new strain onto the farm, because we do see that greater stability in those names. They don't just flip like an RFLP type might.
Yeah. So for a farmer out there or a vet out there that's listening and saying, hey, the bad 144, it would be commonly called, just the bad 144. I got a 144. Is it the bad one? Yeah, it's the bad one. Essentially, that is a 1C.5, right?
That is a 1C5. That's 1C5. Right.
So when people say 1C5, they're talking about the bad 144 in general terms. That would be a pretty quick comparison between what we called it before and what we call it now.
That's exactly right. And the 144 is like the worst RFLP type. Like if you say you have a 144 and you tell me that, I'm like, that could be anything. I cannot tell you at all just on that information what type of virus that is. Yeah. But if you say 1C5, I know that this is part of this group. And it's allowed us to be much more quick in identifying these new emergence events as well, and to have better communication with other diagnostic labs and other researchers that are involved with, PRRS monitoring in terms of identifying the spread of these variants much more quickly than we previously have, because before we didn't even have a unified labeling system that allowed us to identify which farms were part of these, so-called the bad strain that's circulating.
Yeah. So one thing that is very perplexing for me, and this is where I don't want to get in the weeds too much, but one bad 144, also known as 1C5, is a huge group still. It's not like, I just got this little part of my dendrogram or my cluster. It's this little, no, it's a massive chunk of the virus real estate. If the question you asked earlier, is this a new virus or not? Well, it's still a 1C5 this time. I don't know if I failed to get rid of my previous one or if I just never got rid of it or I got infected from somebody new. It's so common out there that I would probably be inclined to say you got to do the actual virus comparison and say, is this 99% like I had, or is this 97 and it's still a 1C5? There's no substitute for actually doing an actual genetic comparison between two viruses, right? Or is that not true?
I agree. The gold standard would still be to do an actual comparison, right? And build your own dendrogram and figure out what else is in your system. Having a label is very convenient for communication, for maybe talking to a veterinarian from a different system and you don't have their data and you can at least say, well, what have you been seeing? But yeah, the gold standard would still be to run those analyses, but they take time and it takes data sharing and it takes someone who knows how to do it, right? And 1C5, it's a very unusual variant to have it take over like it did in, we're talking really 2021, 2022, where it was, and it still is, one of the most prevalent things out there. So we do see farms that have been hit through that 5-year period. They've been hit with this virus a few different times. And has it just always been on the farm, or was it reintroduced? And the other thing that's happened with the 1C5s, I think that they're just a really well adapted virus. We've done some research here led by a graduate student, Marcelo Molini, and he showed that the infectious dose of this particular variant is smaller than other genetic variants, which means that you need less of the virus to infect a pig. Yeah. And what that means is that when we saw that it kind of spread like wildfire in the industry, I don't think it was spreading in new ways. I just think it's because you needed so little of the virus to infect pigs. The biosecurity that works for other variants maybe just was leakier when it came to these viruses that are just, you just need a very, very small quantity to infect pigs. Yeah. So I think that kind of explains why that virus spread so much.
This actually leads into the next natural part of the discussion is, all right, we got all the classification stuff understood. Yeah. But why is PRRS so bad so recently? It seems like, because this 144 has been around for a while, but in the last year, it's like it got a whole new direction on how to destroy the pig industry. It infected farms that it seemingly were a lot safer before. And the question is there more virus? Are they more susceptible to this? Which is what you just suggested, is that you need a much smaller infectious dose. A pig can resist another virus more than it can resist this one, maybe, or is it the amount of virus in the environment, or where is it? What changed? Where is it coming from?
Yeah, so what we've seen, say, in the last, we'll say 8 months at this point, is this group of 1C5 viruses have gotten really large, and we start to see that there's these offshoots that are genetically just a little bit distinct from the original 1C5s we've seen, and so we've labeled some of these genetically distinct offshoot strains now, right? And the one that's spreading really widely right now is the 1C532s. And those ones are now a large percentage of what we're seeing in Minnesota. And probably they have this same adaptation where they can infect pigs with this really low dose, as we saw with the original 1C5s. They probably have that same adaptation, right? Because it's a descendant, it's an offshoot group.
So the 13532 is it seems like that is the super bad one that well, go ahead. But it's like
so two things I'll say relevant for this. So first is that so if you see that middle number, right, followed by the last number. So if this is the 532, the middle number is the parent, and then the last number is the child variant that was an offshoot of the parent. We are not going to do grandparents and great grandparents and make that a really long name where you have 532, 58,
okay,
etc. We are going to leave it as long as it'll get, as there'll be that sub-lineage, which is the 1C in this case, and then there's the parent and then the child number. Okay. And we won't go longer than that. Because we don't want it to get longer, because we know that we developed this to be used by stakeholders in the industry, and they need something that they can easily remember. So it won't get longer than that. And yeah, these 1C532s, many of them still are RFLP 144. So yes, that's one thing to remember, but it would not have allowed us to really identify that this is a separate spreading event. These viruses are upwards of 3% different from the original 1C5s, and they are basically spawning a new epidemic wave in the industry. So having a ID that we now can refer to this group of virus allows us to differentiate these from the previous 1C5s in the industry.
How genetically tight are these 1C532? Is that they're 99.9% the same, all of them, or what's the range that they could even differ?
They've diversified a bit. I would say that you're going to get viruses in that group that are 2% different from each other pretty frequently. I think it's just reflects that group is so prevalent out there. They're just out there evolving like crazy right now. So you're seeing a lot of diversification within that group.
Okay. All right. So we went down the nerd rabbit hole pretty deep on that one. Where did these new viruses come from? Is it recombinant viruses, two viruses mixing? Is it just straight mutation? How do we get new viruses like this that are so bad? Where do they arise? Where do you think they arise?
Yeah, there's two ways that something new, a new strain, can evolve. The first is the more, I guess, traditional evolution, where through time you get these mutations that slowly build up across the sequence. And that is more of a gradual change through time. That sort of evolution doesn't occur in a vacuum. So there are some changes in the genes that have a more adaptive effect of the virus. You're going to see some parts of the genome change more quickly. We actually just completed and published a study looking at vaccine-driven evolution, where we were able to show where we infected vaccinated pigs and non-vaccinated pigs, and we were able to show that the virus changes roughly about twice as much in the vaccinated pigs than in non-vaccinated pigs. So basically what that means is the virus basically is trying to evolve to evade the pre-existing immunity that those pigs have, right? Because the virus wants to survive. So we know that sort of process is going on all the time, and because our pig populations in the U.S. have high levels of immunity, right? Either from previous infection or from vaccination, and the virus is going to evolve to try to escape or evade that immunity. So that's one driver of evolution. The other way that viruses evolve is through a process called recombination. And essentially, recombination happens when a pig is infected with two strains of the virus. And the virus, if it's in the same cell and both viruses are replicating, when that happens, they can exchange some of their genetic material. And so you end up with a virus that looks a little bit like virus A and a little bit like virus B, and it's like this mosaic virus, and we call that process recombination. And this does happen a lot. It particularly would be likely to happen in farms that are using a lot of LVI, particularly if they're using multiple different strains to inoculate their pigs. But it also could happen if we're using live vaccines, like the MLV vaccine or any of the modified live vaccines, in a herd that has an active outbreak of PRRS, right? Because those viruses are now mixing in those animals. And so this is a more rapid shift in evolution, because they can, change a large part of the genome at once, and you get a new virus that looks a little bit like both parents. And we do know that some of these new variants, like the 1C5s, had a recombinant origin, and sometimes recombination results in these really fit viruses. Sometimes it may not. It really just depends what happens in that animal. But certainly it is a way that virus or new variants can emerge.
Kim, how do you, that you mentioned that you said, oh, these 135 group had some recombination with another virus that led to this. How do you know that? Is it a whole virus genome sequence that you have to do? How do you determine that this one came from a couple that got mixed versus this one just slowly changed by itself?
Yeah, there's two ways, and this might be getting into the weeds of the nerdy part of this. One thing is, so the recombination doesn't always involve OR5, so you cannot always see it just by looking at your OR5 sequence. But these 1C5s, the recombination is not in OR5, but when you looked at those early trees, right when their emergence happening, they were on this really distinct long branch of the tree, kind of out on their own. And it's because that all of those viruses had that same ancestor that had this recombination or recombinant origin, and then they just started evolving on their own trajectory. But generally how you would do this is with a whole genome sequence, and you would need a whole genome sequence and compare it to other whole genomes to really be able to determine when recombination has happened.
Okay. Hey, other question, kind of related. Where do new viruses come from? Probably somewhere where there's just lots of pigs and lots of virus circulating because of recombination that could occur. Is that a fair statement, or am I off base on that?
Yes and no. I mean, I don't want to point fingers at any one region, but the diversity of PRRS is greatest in kind of this upper Midwest area, right? There's just more pigs, there's more PRRS. All roads lead to the upper Midwest.
Sioux County, Iowa is what you're saying. You're blaming the Sioux County, Iowa farmers.
Right. We are bringing pigs in from all over, really, the continent into the upper Midwest, right? So all of the strains that are in everywhere in the country end up here in this mixing pot of Iowa and southern Minnesota. Yeah. So there's a lot of potential for recombination to happen here, and then there's just a lot of evolution happening here.
Yeah. Okay. I'll quit picking on Iowa, but I think it seems clear that when you have lots of pigs from lots of places, that's where new PRRS issues seem to arise. For like a set of viruses originate somewhere down there. That's where most of the pigs are. Hey, we've talked a long time, and we've burnt up about half an hour. And we could talk a lot more on this, and I apologize to our listeners for getting into the weeds too much. But I think most of the people out there listening would say, yeah, I've talked to my vet, or I've looked at these lab reports, and I'm used to talking about them as 144, and now there's these different numbers, which you just explained. It's this lineage, sub-lineage, parent-child coding, which if you get used to talking about that, it'll add a lot more precision to the conversation versus, well, which 144 did you get? Well, I don't know. You'd have to compare it. This at least lets you talk in terms that can compare that more accurately, correct? Is that well said?
That's very well said.
Okay. And then we talked a little bit about where viruses originate and how they arise, and the current bad strains that are out there. And you've talked about slow, just mutation that may not be slow, but may be driven faster if they're vaccinated, or when two viruses recombine and you get a whole new kind of a Frankenstein virus that's got some of both that show up as a new virus. And I think I'll just leave it at that. I really appreciate your time. If any listeners want to learn more about this, you can contact any of the vets here within our group, because we'll have a real cursory knowledge of this new system. But Kim, I'm going to throw your name out there too. Do you do extension work? I don't know how that fits in with what your daily routine is, but Kim VanderWaal is a professor at the University of Minnesota, and if you could probably find some stuff online that you've already published regarding the new classification, I'm guessing.
Yes, we have a paper which we tried to make it relatively lay oriented, or at least showing the applied use of this system and trying to demonstrate how the new nomenclature can help track viruses from kind of the farm perspective. And then we also have a website where you can classify your own viruses there. But we also there's like a fact sheet there that has kind of the quick bullet points on what we talked about today. So you didn't catch it all. It's one-page bullet points. So
Kim, where can they find that? Because I want to look up that compare your own virus or classify your own virus thing. Can you?
Yeah, so that's the PRRS Loom website. I will drop you the link, and maybe you can attach it to the show notes.
Okay, PRRS Loom website. Okay, I'm going to look it up because this actually came up today in conversation. Somebody wanted to know which virus they had, and I said, I don't know, ask him. But if there's a website we can actually drop it into and get that done, that's even easier.
I'll send it your way.
Okay. Thank you, Kim. And thank you to our listeners. Hopefully we didn't get too far into the weeds on this, and I know we did, but it is good stuff, and it's a lot to think about, and people are thinking about this. So I really appreciate, Dr. VanderWaal, that you were here today. Thank you.
All right. Thank you so much.
The SwineTime podcast was created for the pork industry and individual pork producers around the country. Hosted by me, Dr. Spencer Wayne with Pipestone Veterinary Services, the podcast contains pork industry news, advancements in animal care, and how to enhance your productivity. Find the SwineTime podcast at www.pipestone.com or on your favorite listening platform.